Hi, I'm Alyssa Playa, and I'm here today with my group mates to talk to you about our paper, Durable Contraception in the Female Domestic Cat Using Viral Vector Delivery of a Feline Anti-Malarian Hormone Transgene. To give you some background, this study focuses largely on the inhibition of follicular genesis through superphysiologic levels of anti-Malarian hormone. A previous study performed by the same scientist in mice found that superphysiologic levels of anti-Malarian hormone introduced by an adeno-associated viral vector transgene inhibited the development of primordial follicles into primary follicles and primary follicles into secondary follicles, meaning both the gonadotropin-independent component of follicular genesis and the gonadotropin-dependent component of follicular genesis was inhibited, indicating to them that this could be a robust method of contraception. So this led to the question if a single administration of this adeno-associated viral vector to overexpress anti-Malarian hormone could provide a safe and effective, durable contraception method for female domestic cats, potentially offering a solution to the feral cat overpopulation. The hypothesis they put forth was that an adeno-associated viral vector-mediated delivery of a feline-specific anti-Malarian hormone transgene will suppress ovulation and prevent pregnancy in female domestic cats, offering a non-surgical and long-term contraception method while allowing normal hormonal cycling. Who broke this paper's experimental setup into transgene design and animal models. So we have an example of the transgene of their version 2 transgenes shown down there, and I'm going to get into some of the animal models used in this study. As mentioned previously, kind of a proof of concept was performed in a mouse model, and this current study uses cat models with a pilot study showing proof of function of anti-Malarian hormone delivered by an adeno-associated vector, preclinical study that looks at 2 doses of our transgene in that vector, and a control, and both of these studies were performed over 2 years. So, kind of to tailor transgenes to the domestic cat, the researchers created 2 generations of our feline anti-Malarian hormone transgene, as seen here, with FC standing for Felis catus, MIS standing for Malarian inhibiting substance, which is another term for anti-Malarian hormone, and our version 1 and our version 2. Version 1 was generated using a feline genome consensus sequence of an earlier version, version 0.8.0, which was available when the study first started. And it actually had significant gaps in it, which had to be filled in using dog consensus sequence. Version 2 came along after the pilot study, and it used our feline genome consensus sequence, version 9.0, which had the full anti-Malarian hormone gene present. And they actually found between the 2 versions there was a 31 amino acid difference between the two, which could explain some of the immunogenic responses we saw in the pilot study. Both transgenes were then codon optimized for feline expression, meaning that any codon bias displayed in the feline species was then selected for, and both of our versions were engineered to reduce their GC content using a GenSmart algorithm. Next, the transgenes were then tested for their functionality using a fetal rat urogenital ridge bioassay, which is shown here. And our transgenes were then tagged with a flag tag and transfected into mammalian expression vectors and transfected into mammalian cells. When these cells then express these proteins, the flag tag allowed them to be purified out of our culture and then tested on these urogenital ridges, where we are looking here at this significant structure called the Müllerian duct, which when functioning anti-Malarian hormone is present, should regress, as we see here in comparison to our untreated versus our treated. That Müllerian duct has significantly regressed, showing that they were successful. Finally, to then incorporate this transgene into our cats, it needed to be inserted into an adeno-associated virus packaging plasmid, and these packaging plasmids contained a strong CMV enhancer, a strong promoter, a synthetic intron, and a polyadenylation signal, which all just increase the expression of this transgene once transfected into a cell. Now we're going to look at the 3-cat pilot study. The cats were between the ages of 6 to 7, and they were given only 1 high dose of our version 1 transgene. And measured through capture ELISA, we looked at the anti-Malarian hormone antibodies present in the serum and the amount of anti-Malarian hormone present in the serum as well. And they found that there was a varied response between the three cats. This first cat, named Antilles, had the best response to it, where he didn't develop any detectable anti-Müllerian hormone antibodies. In Catalina and India, they both had a robust antibody response to it, and India had a complete absence in the production of anti-Müllerian hormone and a very strong antibody response. Just to kind of confirm that this injection was safe in all of the cats, they looked at C-reactive protein, which is an inflammation marker, and they showed that after an initial increase following the injection, these levels dropped low enough that they felt that it was safe to continue. 2 years into the study, they then spayed the cats and removed the ovaries and analyzed them for evidence of follicular genesis. And they found for Antilles, who the initial version 1 worked the best, they did not see any signs. But in Catalina, you begin to see the presence of antral follicles shown here in the using the black arrows. And in India, we see the sign of a corpus luteum, meaning not only have these follicles developed, they have also ovulated. Sadly, 2 months after the study, India was struck by a car, and her body was then donated to the scientists for necropsy, where they then performed in situ RNA hybridization to assess how that anti-Müllerian hormone transgene was being expressed in her tissue, even with that strong antibody response. And they found that in the liver, there was a reduced expression of that anti-Müllerian hormone, and they thought that might have been due to the high cellular turnover of the liver in comparison to our skeletal muscle, which acted as a reserve and continued to produce anti-Müllerian hormone over that 2 year study, and they felt it could be used for longer term applications.
So with the second version of the transgene, there was success in creating or in increasing the anti-Müllerian hormone secretion in cats. After the pilot study, this new experiment was done with the, as mentioned, the revised transgene. It was done on 3 groups of cats, each with 3 groups receiving high doses, low doses, and control doses of the transgene. Um, this was injected into the thighs of the cats. They did some well-being assessments, checking for any pain behavior, increases in temperature of the cat's tissue mass formations, changes in physical or blood levels of various things. And they were assessed for 2 years. Um, Again, as mentioned, the anti transgene was measured with ELISA, and no antibodies were detected, which was great. It was a good sign indicating that the cats were not developing a immune response to the transgene. And then shown by the figure to the right, the circulating anti-Müllerian hormone levels did decrease throughout the first year and ended up plateauing in the second year. However, this was still great because the levels remained above the 0.25 micrograms per milliliter, which was the target concentration.
After verifying that AMH secretion was sustained in cats that received the treatment, the researchers wanted to investigate how the levels of other reproductive hormones might be affected. To do this, they tested all the cats used in the study for multiple hormones across 3 different time periods. Most hormone samples were collected from blood serum 6 months prior to treatment, called the pre-treatment phase. During the 2 months following treatment, this was called the transition phase, and then also 2 years post-treatment, or the post-treatment phase. Hormone concentrations were determined with ELISA test kits or also some modified enzyme immunoassays. Changes in hormone levels were then compared between the 3 different treatment groups, the control, the low dose, and the high dose group. Estradiol and progesterone sampling was conducted slightly differently than some of the other hormones. These hormones were collected from fecal samples 3 times per week throughout the entire duration of the study. This high-frequency sampling was designed to capture the appearance of the estrus phase and luteal phase, which are important stages of the mammalian menstrual cycle, to more directly observe the physiological changes in the cats. When conducting the hormone studies, the researchers found that hormones associated with preantral and antral follicles were not affected by the elevated levels of AMH. These included inhibin A, inhibin B, follistatin, estradiol, and testosterone. The appearance and frequency of the estrus phase, which is defined as a period of sexual receptivity in mammals, was also not impacted by the treatment. This was determined by observing elevated estradiol as 1.5 standard deviations over the mean for 2 or more consecutive samples. This finding was promising because it demonstrated that estradiol, a hormone with multiple beneficial health impacts on cats outside of reproduction, would not be compromised by the treatment. When analyzing changes in later stage reproductive hormones, the researchers found significant differences between the treatment and control groups. Treated cats exhibited elevated post-treatment luteinizing hormone concentrations and reduced progesterone, a phenotype that is consistent with the hypergonadotropic hypogonadism condition, in which pituitary hormone levels are elevated as an attempt to compensate for impaired gonad function, and there's little to no sex steroid production as a result of this. The reduction in progesterone concentration causes ovulation suppression, one of the key contraceptive features of the AMH transgene treatment, although the authors do point out that further research is needed to verify the exact hormonal mechanism behind all this. In addition, this hormone imbalance directly impacted the appearance of the luteal phase, the stage at which the uterine lining thickens in preparation for a fertilized egg in cats treated with a high transgene dose, but not the low dose.
So a mating trial was conducted to test the efficacy of the treatment. This was done twice, each time with a 4-month trial. In this trial, a male cat was placed into a home with the 9 other cats for 8 hours a day, 5 days a week, and all interactions were recorded with a little videotaping. And then these films were analyzed and scored as successful breeding or breeding attempts. And with this, pregnancy status was assessed via ultrasound weekly. The results are shown here to the right. It's very interesting. Only 2 of the treated cats participated in any type of breeding activity, whereas the control groups had a lot of breeding. They gave birth to many healthy kittens, whereas the cats with both low and high doses of treatment had no gestation. Yeah. And then some issues with the study. So the sample size was fairly small, each group consisting of only 3 cats. This might limit the statistical power and generalizability of the findings. Another flaw, the cats were only monitored for 2 years post-treatment, so we weren't really able to look into the long-term health of any reproductive organs, like development of uterine conditions, things like that. And the opportunity to look into internal components could not be assessed when the cats are still living. So like this might require some sort of necropsy, which wasn't done to the cats. Well, except for one of them, but in the pilot study. Anyways, and then another issue, the mechanism by which the anti-Mullerian hormone suppresses ovulation is unknown. The paper had the interpretation that the conception observed is caused by a direct effect on anti-Mullerian hormone on the ovary, which would inhibit ovarian follicle maturation and ovulate in response to an LH surge. However, they were not able to rule out the possibility that contraception was just due to an LH surge alone. And then one final issue, there were some potential confounding variables. C-type natriuretic peptide and follistatin, which are hormones, changed only in the control groups, but not in the treatment groups. So it's kind of unclear whether this result would be influenced by anti-Mullerian hormone.
In the end, the researchers have shown how the intramuscular injection of the transgene treatment results in sustained secretion of anti-Mullerian hormone at supraphysiological levels, denoting a durable contraceptive effect. They pinpointed the most likely explanation for contraception as the inhibition of breeding-induced ovulation, although future studies will need to build upon these initial findings. Looking at the bigger picture, this treatment is particularly promising because it is efficient, it's marketed as being cost-effective, and better for the health of the cats than an ovariectomy. There's also evidence that the chances of life-threatening cystic endometrial hyperplasia, or pyometra, is reduced by the anti-Mullerian hormone transgene treatment through the prevention of spontaneous ovulation. And lastly, the fact that most sex steroids are unaffected by the treatment is a promising indication that cats receiving the transgene will retain the positive health benefits of these hormones and their longevity will not be impacted.